Good morning. I'm Keith Yamamoto. I'm a molecular biologist at UCSF and also serve as the Vice Chancellor for Research and the Executive Vice Dean of the School of Medicine. And it's my distinct privilege to welcome all of you to the 2014 Breakthrough Prize Life Sciences Symposium. I'm sure that most of you, if not all of you, know that the Breakthrough Prize was founded in 1913, designated on the life sciences side as awards for discoveries that extend human life, is what it said. And really what we all recognize is that really fundamental, bold research directions and discoveries are the fuel for discoveries that extend human life. The awards are sponsored through generous grants from a board chaired this year by Corey Bargman and comprised of Bay Area entrepreneurs, very generous ones, Mark Zuckerberg and Priscilla Chan, Sergey Brin and Ann Wojcicki, Yuri and Julia Milner, and Jack Ma and Kathy Zhang. And so we owe them a great debt of thanks for both designating the awardees, funding the awards, and providing the reason and excuse for all of us to be gathered together. So let's begin by giving them a big hand. So today, in this Life Sciences Symposium, we have an incredibly exciting day of science in store for us. Thanks to the efforts of a planning committee from the three host institutions for this event, Stanford, UC-Berkeley, and UCSF, and from the Milner Foundation. And of course, a big thank you to Stanford, which is our host venue for this year. This is a rotating symposium on a year-by-year basis. So our exciting day is exciting in many ways because of the broad base of talent that it will draw from, including many of you in this room. 5 of the 6 2014 awardees, Breakthrough Prize awardees, are with us and will be presenting talks. Then we'll have a panel discussion with the 2015 Breakthrough Prize awardees, and importantly, a group of students and postdocs from Stanford, Berkeley, and UCSF. And then over the lunch break, we'll all have a chance to go out into the sun and enjoy posters and interact with postdocs and students who have generated exciting work that we'll be able to view and interact with them about. So I think that to my view, an exciting element of this day is not only that we've been able, through the Breakthrough Prize Foundation, to pull together scientists of such demonstrated talent and innovation and boldness and skill, but I would venture that maybe the smartest people in the room are those that are going to be the students and postdocs that will be involved in the panel discussion, asking questions of the new awardees, those who are presenting posters, and some of you in the room who are attending. And I think that's probably, statistically, it's not a bad bet that that's where some of the smartest stuff is going to actually happen, despite the demonstrated prowess of the awardees. So it's great to have everyone together and pull together in this way. So our speakers in this first session are loosely connected by having done time in Bay Area institutions. And they've also had, from different points of view, some foci on T cells and their functions. So let's just get right into it. Our first speaker is Dr. James Allison. Jim received his PhD in 1973 from UT Austin and carried out postdoctoral work at Scripps. I first met him, I think, around 1980 or so, while he was an assistant professor at the MD Anderson facility in Smithville, Texas, where I attended what turned out to be a remarkably raucous meeting, I want to say. Jim's link to the Bay Area was in Berkeley as professor of immunology and director of the Cancer Research Laboratory beginning in 1985, where he was jointly appointed at UCSF as an HHMI investigator as well. In 2004, Jim moved to direct the Center for Cancer Immunotherapy and chair the immunology program at Memorial Sloan Kettering Cancer Center in New York. And in 2012, he returned to MD Anderson in Houston, where he is chair of immunology. Throughout this time, Jim has been at the leading edge of investigations of T cell development and activation, and the concept and strategy of tumor immunotherapy. In 1996, Leach, Crummell, and Allison published a landmark paper in Science showing that antibody blockade of a T-cell inhibitory molecule, CTLA-4, could lead to enhanced anti-tumor immune responses and to tumor rejection, sort of the leading edge of this concept of tumor immunotherapy. This concept of blocking T-cell inhibitory pathways as a way of unleashing anti-tumor immune responses laid the foundation for the development and eventually FDA approval of a drug, Yervoy, for treatment of metastatic melanoma and subsequently to other such immune checkpoint therapies. So Jim is a member of the National Academy of Sciences and the Institute of Medicine, a fellow of the American Association for the Advancement of Science, and in addition to the Breakthrough Prize in Life Sciences, he has received many other awards that I will not enumerate. Please join me in welcoming Jim back to the Bay Area for his talk, Immune Checkpoint Blockade in Cancer Therapy.
Try to present this from the podium. It's a little hard to see the slides, though, so I'm going to try to, at least initially, see if I can point with the pointer here. Anyway, I want to thank Keith for that very kind introduction. It is really great to be back here. I did actually, in addition to my time at Berkeley, I did a year sabbatical in '82 and '83 here at Stanford in Irv Weissman's lab. Learned a good taste for science and fine wine during that period here, and as any of you who know Irv will know for sure. And then actually towards the end of my time at Berkeley, I also had a joint appointment at UCSF, so I've had appointment at all three of the places here. So what I want to do today is just talk to you about what Keith just introduced, immune checkpoint blockade in cancer therapy. And what I'd like to do is start off with a really brief introduction, trying to put this into context of cancer therapy, and then really try to concentrate, as we were asked to do, on where I think this field is going. Has gone in the last year and is rapidly going. And you'll notice the title on the slide is Prospects for a Cure, because I think we can confidently say, and I hope you'll agree with me at the end here, that we're within reach of actually being able to cure many kinds of cancers, at least if you define a decade or longer survival after a single round of therapy as a cure. So why would you want to do immunotherapy? Well, there's three features that the immune system offers that no other modality of cancer treatment offers. The first is specificity. T cells, as many of you know, I'm sure, recognize peptides that are sampled for basically everything that's going on in a cell. MHC molecules take them to the surface. T cells come by and look for things that oughtn't to be there and then mount an attack. And so this is very effective in dealing with cancer. But what we know now, work in the last few years, something was initially proposed by Bert Vogelstein and I about 8 years ago, was that there's so many mutations in cancer that have the potential for generating neoantigens that it might be that it was just these neoantigens themselves that are the targets of the immune response, that the immune system will treat them like a virus infection. And so we now know that that's correct, and that those are the major targets. And so that being the case, if you look at it that way, what the immune system does is target the very process of carcinogenesis. It targets the process of mutagenesis and genomic instability that's a fundamental feature of cancer. The second thing is, once you've got T cells that recognize these things, you've basically got them, or memory cells generate from them, which are slowly self-renewing cells for the rest of your life. And so if the tumor peeks its head up again, the T cells there, they can take it out again. And the final one, and perhaps most important, is adaptability. I think everyone knows of the incredible adaptability and mutability of tumors, particularly large tumors, as a result of the large number of cells, all of which are genomically unstable. There's incredible heterogeneity in the tumor. But there's one thing that the immune system loves, that's heterogeneity and new antigens. In fact, the immune system has its own process of somatic mutation, which is harnessed and regulated very tightly, that can generate, according to Mark Davis here at Stanford's calculations, as many as 10 to the 15th power different antigen receptors. Obviously many more than you have in your body, but you have probably 100 million different T cells running around in your body that can recognize different things, because the whole system is designed to protect you against anything that nature throws at it, including, I would argue, cells expressing new peptides generated by mutational events associated with the carcinogenic process. Well, that being the case, then why hasn't immunotherapy worked in the past? There have been decades of attempts to actively engage the immune system. We know that there are T cells in human tumors. We know what the antigens are. And so people have been trying therapeutic vaccinations for decades. There have probably been close to 700 different clinical trials trying to do this, and they've never worked, really. I mean, there have been some anecdotal responses, but as a general modality of cancer, it's never worked. And so that puzzled many people to the extent that it became embarrassing to be somebody who was interested in cancer immunotherapy. In fact, it was kind of a pejorative in the late 80s and early 90s when somebody, my friends that were fundamental immunologists, they introduced me and said, well, Jim, he's a tumor immunologist. Anyway, shudder a little bit. But I wasn't really a tumor immunologist. I was a basic immunologist trying to study, as Keith mentioned, I've always been very interested in what regulates T cell function and activation. It turns out it's a much more complicated process than we thought. After the discovery of the antigen receptor in the early 80s, it was thought that all you needed was engagement of the antigen receptor by a peptide MHC complex on an APC. But it was shown very, very clearly by Mark Jenkins and Ron Swartz at the NIH that it was a lot more complicated than that. There are additional signals called costimulatory signals that could only be delivered by professional antigen-presenting cells, dendritic cells, cells that Ralph Steinman got the Nobel Prize for discovering a few years ago. So these dendritic cells have these costimulatory ligands, which we now know are molecules called B7, B7-1 and B7-2, and they engaged some receptor on T cells, which we showed in the late 80s, early 90s, was a molecule called CD28. And many labs went on to show that when a T cell gets contemporaneous delivery of the antigen receptor signal or signal 1 and costimulatory signals mediated by CD28, signal 2, you turn on all this stuff and the cells start headed towards cell cycle progression. You turn off inhibitors of cell cycle progression, which lets them go. You induce anti-apoptotic factors, and then the T cell can start dividing very, very rapidly and differentiate to effector function and deal with whatever it needs to deal with. But when we cloned mouse CD28, there was another molecule that had already been identified by Pierre Goldstein in France. He didn't know what it did, but he knew that it was present in all T cells after activation. And so anyway, there's a lot of controversy about what it did. It was shown that it binds the same ligands as CD28. And so early on, there was a lot of thought that it was actually another costimulatory molecule. And Peter Lindsley at BMS published a number of papers saying that. But by 1993 or so, Jeff Bluestone, who was here earlier, was at Chicago at the time in my lab, began to doubt that. And we decided that that wasn't what it did, that it was actually an inhibitory molecule that actually served to slow down, if not stop, immune responses, and that antibodies to it looked like they were stimulatory because they were removing a negative signal. Now, this was very heretical at the time because it was thought that, however you activated a T cell, one signal, two signals, it didn't matter. If they died by activation-induced cell death by fast ligand interactions, and that's what terminated them. But anyway, Jeff and I proposed that CTLA-4 was actually a negative regulator. And it's interesting when you put this in context with all the biology that we know now. Again, this binds the same ligands as CD28, binds them actually 2 logs at least tighter. And so that kind of gives you a bad feeling for whether it could actually be a negative molecule, because that means that the off signal is inherently hundreds of times stronger than the on signal. And the way the T cells solve that problem is that CTLA-4 is not present in resting T cells. It's induced by the program that started when you get co-stimulation with CD28. That activates the CTLA-4 gene. The T cell begins to make this inhibitory protein, which can then bind up the same molecules that bind to CD28 and basically stop co-stimulation and just stop the whole process. And basically the T cell would run down and have to stop dividing after that. So the idea is that this is a program that's inherent in T cell activation. You have to stop T cells or they will kill you. In fact, several years after this, Tak Mak and Arlene Sharp and Cynthia Chambers in my lab knocked out CTLA-4, and the phenotype of the mice is they basically turn into a giant lymph node when they're about 3 weeks old because their T cells can't stop dividing. It's not really autoimmunity, but the T cells just fill up all the organs. They start making cytokines and they die of a cytokine storm. Anyway, it's fascinating biology, but for the purposes of this talk, I'll just say that it occurred to me when we made that observation that maybe this is why therapeutic vaccines didn't work. So there's two issues about this. One is something that we noticed in several years before we started this work, is that solid tumor cells never express the costimulatory ligands. So they're invisible to the immune system until the tumor gets big enough to block off the food supply or outgrow the supply of oxygen or something anyway, and then start dying, cause inflammation, attract the dendritic cells which come in and pick up the bit of dying tumor cells, represent them in the context of B7 molecules on the surface of these dendritic cells, and then you get co-stimulation, you initiate immune response. But CTLA-4 then is induced, and by then we would argue that the tumor is so big that the T cells can't take care of it before that hardwired program catches up and says, stop, don't kill these cells because they might be self. You know, there are too many of them and we shouldn't kill them. And when you start vaccinating, particularly if you start vaccinating regularly, you've already started that program. So after the first couple of injections, when you re-inject, you're just hitting the off signal. Because every time you give an antigen receptor signal, that tells the cell to make more CTLA-4 until finally the process runs down. So we had the idea that that's how it worked. So that we could deal with this by something as simple as monoclonal antibodies to CTLA-4, which would prevent their interaction with B7, keep them out of the picture. You could suspend the brakes, basically, for as long as you want, because the antibodies, you inject them, they can be cleared. But you could just suspend the brakes and let the T cells keep running for longer than they would have normally, and hopefully take the tumor out. And so this was a very compelling, when we first thought about it, for a couple of reasons. One is, if you look on the right side of the slide, there's no tumor cell there. It doesn't matter what the tumor is. It doesn't matter what drives the tumor. It doesn't matter whether it's kidney, melanoma, or whatever by this model, because the tumors will have antigens. What matters is that you're interfering with the immune system. So you're not treating the cancer. You're treating the immune system. So it's not a cancer drug. It's an immune drug. And the second thing is, you are not trying to turn it on. You're just trying to keep it from turning off. So those were two pretty radical ideas at the time. First, treat cancer by ignoring it. And the second one is don't try to turn the immune system on, try to keep it from turning off. So you're not harnessing it, you're unleashing it. And so anyway, those ideas were compelling. It could offer the chance of being a universal cancer drug. But the other is you could use it as monotherapy to take advantage of the priming that occurs when you have necrotic cell death. You can make vaccines work better, but also use it in combination with virtually anything that kills cancer, kills tumor cells, because that will start this cross-priming. It'll just be terminated too early unless you do something about CTLA-4. So does it work? Well, this is our first experiment, and luckily we picked the right model, because here you can see the tumor grows. If we inject antibodies to CD28, the tumor grows faster, showing that you need CD28 to get an immune response. There is an inherent immune response, but it just can't take the tumor out. But if you inject antibodies to CTLA-4, the tumor grows for a while, and then the tumor gets rejected. And the mice are permanently immune to re-challenge with that tumor. And so this was pretty exciting because, I mean, it was worked even better than I thought, because all we're doing is covering up one molecule out of everything that's going on when the host responds to a cancer and the cancer responds to the host. Just taking this one signaling pathway out is enough to turn death of the mice into permanent immunity. And so we then proceeded to a lot of other tumor models. And what I can tell you is if in strongly immunogenic tumors, monotherapy worked, but against poorly immunogenic tumors, it didn't work. This shows B16 melanoma, which is very poorly immunogenic. Anti-CTLA-4 really does nothing. Glenn Dranoff had made a tumor cell vaccine that expresses GM-CSF, the cytokine which enhances cross-priming and is a chemoattractant for those dendritic cells and upregulates the costimulatory ligands. So we felt, and he showed that it worked prophylactically against B16, but it never worked therapeutically, which we confirmed here, as you can see. But it seemed the perfect pair to get together because GM-CSF gives you all the stuff that starts an immune response that CTLA-4 then could sustain. And as we might have predicted from this mechanistic understanding, when you put them together, 0 plus 0 is 100%, and you cure essentially all the animals. And again, they're permanently immune. So I'm not going to go through any more mouse stuff except to say that we have never found any model that we couldn't treat successfully with some combination, with be it chemotherapy or radiation or a vaccine like this. So what goes on? Well, this is that B16 tumor untreated. You can see the vasculature here in red, the odd green cell out there that's a T cell. If you look at the kinds of T cells, a lot of these guys have nuclear FOXP3. For the non-immunologist, that's a transcription factor that tells that cell to be a suppressor cell. It makes TGF-beta and IL-10 and really keeps effector cells from coming into the tumor. But when you give anti-CTLA-4 in the vaccine, you can see now the vasculature is activated, expresses the integrin, ICAM in this case, also VCAM, which allows the T cells to stop and extravasate in the vicinity of those molecules. They go out. Now you look at the count of T cells, there's still some of those regulatory T cells with the purple nuclei, but there's a lot more FOXP3 negative CD4s and CD8s. And if you sample the cytokines, there's no IL-10 or TGF-beta anymore. It's all TNF-alpha and gamma interferon, which is an inflammatory environment. The top panel, this mouse will have to be euthanized within been about a week of that time that's made. This mouse, about a week or so later, will have no tumor, just a depigmented spot where the tumor used to be. In any event, we teamed up with a company called Medarex that had mice that made where their genes, the immunoglobulin genes, had been replaced by human, made a fully human antibody called ipilimumab. Actually, just a joke for a moment, when we started this project, it was called the Medarex project, the 010 project. It was their 10th antibody, so we called it the 010 project. The FDA named it ipilimumab, which is a kind of a nonsensical name, but I was at Berkeley at the time, and I told Medarex they ought to at least go to the FDA and have them put an H in front of it. And, but they didn't think that ipilimumab would be a good name for a drug, I guess. But in any event, so there have been over 50,000 patients treated with this drug now that have been objective responses in many tumor types, just as we would have thought, including a lot of different kinds of cancer. There are some adverse events. But this is one of my favorite slides. This is a woman, on the left is her condition in 2001. You see this tumor, this melanoma metastasis here in her lung. 6 months later, after a single injection in a phase 1 trial, her tumors were away. This is a CAT scan 10 years later after that single injection, and her tumors are still gone. And that was 3 years ago. She's still doing fine. You can see this woman had these tumors on her legs. She was treated by Jed Walchuk at Memorial. At baseline, you can see all these tumors. The standard treatment is 4 doses at 3-week intervals. You can see by 6 weeks, the tumors are mostly gone. By 10 weeks, they're completely gone. So sometimes the patients don't respond quickly enough. You can see this patient's tumors got bigger, got a new lesion, but eventually all the tumors went away. And this is just an example here of prostate cancer. You can see the bladder here compressed by this big prostate cancer mass. After being treated, the tumor's completely gone. In any event, this is then back to melanoma. This is an 800-patient randomized placebo-controlled trial of metastatic melanoma. All you need to concern yourself with is the top line, which is anti-CTLA-4 only, or the bottom line, which is the placebo control. You can see the median survival moves over by about 4 months. But you can see here the tail flattens out at about 2.5 years. At about 22%, it stays there for the duration of the trial, which was 4.5 years. So this tail was pretty exciting because it means, the patients that made it 2.5 years or so made it through the trial. But there was recently a 5,000 patient follow-up by Steve Hody that showed that the patients that made it that long were still alive at 10 years. So a patient that makes it 2.5 years still is going to be alive 10 years later. So basically nobody dies of the disease. CTLA-4, we know now, is not the only checkpoint. In 2001, Arlene Sharp and Gordon Freeman together with Tasku Hanjo showed the PD-1 pathway, another molecule that works different. Well, I'll show that in a minute. It also has activity against melanoma. These are spider plots with individual patients responding. You can see about 30% of the patients have objective responses. They're another promising drug. But these molecules work differently. Like CTLA-4, the ligands of PD-1 are on antigen-presenting cells, but it has one of its ligands can also be on tumor cells. So it's a little bit different there. And it's also different in that it interferes with T-cell receptor signaling, not co-stimulation. Completely different mechanism. And so that led us to think they might be additive. And so in mouse models, we showed that they are. You can block them together, and it works better. That led to a clinical trial last year. in which the response rate, with 65% or 2/3 of the patients had tumor shrinkage. Half of them reached objective levels, that means 50% or more tumor shrinkage. Of those, half had 80% or more tumor shrinkage. And so now we're up to a 50% objective response rate, except there's one thing worth pointing out. With anti-CTLA-4, the objective response rate was 8%, but 22% lived for 10 years. So I think you'd rather go for survival rather than the objective response rate. So this trial then has been followed up with considerable interest. There are actually several doses, but if you look at this line here, this is the optimum dose with 1 mg per kg of anti-PD-1 and 3 mg per kg of anti-CTLA-4. The 2-year survival here of all comers is 88%. And if you look at the trial earlier with the placebo control, at the end of 2 years, only about 7% of the patients would still be alive. So this is a pretty remarkable thing. If, and again, it's a small number of patients, it was not a randomized trial, so we don't know what it's going to be like, but hopefully it'll be somewhere upwards of 50%. So this disease then, 5 years ago, would have been a death sentence in about a year to a year and a half for the vast majority of patients. Now we have 90% of them surviving at least 2 years, and if they make it another 6 months to another year, they're going to make it for 10 years, is what we would argue. So there are a lot more of these molecules to look at, and I'll just show you this hairball here. We've got a lot of work to do. For those of you who are doing postdocs or just starting out, we need a lot of help. There's a lot of different ways to go on this. So, but where we're going to go now are multiple checkpoints, combining things. I don't have time to show you, but we have recently identified a costimulatory molecule, ICOS, which you can target together with CTLA-4 to get a considerable improvement in efficacy in animal models. We're about a year away from having that in patients. A lot of other things that we can do, including genetically targeted therapies. I'll just show you here one possible therapy that looks really promising. This is Sutent, a TKI, that's the standard of care for metastatic renal cancer, has about a 35% response rate. Here, combined with the antibody to PD-1, every single patient shows some tumor shrinkage in that. Now, we know nothing about the durability of this yet, but again, every single patient shows a response here. And just to close here, we know now about tumor antigens. Again, just to come back to what I started with, there's a lot of tumor antigens that are in normal cells as well. But these that are generated by single point mutations, deletions, frame shifts, fusions, or whatever, we think are what the immune system's focusing on. This just shows the range of tumors with melanoma and smoke and lung cancer and people that smoke have several hundred to thousands of point mutations, or just mutations that can generate new peptides. And this ranges across many numbers. We know that at least down as low as prostate cancer and renal cancer, there are responses. So while there is a correlation perhaps with response and mutational load, that's not absolute. We can go pretty low. So one of the things that we're trying to do now to move the field forward is to develop another kind of personalized medicine where we do genomic and then sequencing of the patient and then RNA sequencing of the tumor, identify those peptides that can be recognized by the immune system, and then develop vaccines which can be personalized for the patient and given with the checkpoint blockade to really try to get this to work in patients that may not respond to just the checkpoint blockade by itself. So finally, I'd just like to say that, we've been used to cancer therapy moving the median survival over a little bit. We know from anti-CTLA-4 that you can get a tail on the curve at about 20% that'll last for a decade. And so we know from when you combine it with PD-1 or perhaps a checkpoint inhibitor, we can move that up to somewhere between 50 and 90% if those numbers again hold on the trial I showed you. That's melanoma, and so our task now is to really extend this to many other kinds of tumors and other combinations to see if we can't really, again, forget about this and start concentrating on curing cancer in the majority of patients with a large number of tumor types. So I'll stop there. Sorry I went a little bit over. Thank you.
I was wondering if you could comment on strategies to approach, using this kind of therapy on some of the tumors that you noted earlier that are non-antigenic, and for some reason they're not particularly antigenic to begin with. And how do you approach trying to reach that?
Lung cancer was thought to be non-immunogenic because nobody had ever shown it to be, but it responds with about 40% rate to monotherapy with PD-1. And so I would argue that all tumors are inherently immunogenic because they will have mutations. But I would also say that sometimes the mutations that might not generate antigens of high quality, in fact, Bob Schreiber has shown this in mouse models, that the immune system won't see unless you give anti-CTLA-4, with a vaccine, and then you can reveal them. But I think another factor is that some tumor cells might have multiple checkpoints involved, so they appear to be non-immunogenic. But what we already know from these sorts of studies is that patients that don't respond to anti-CTLA-4 will respond to PD-1, and vice versa. And as I showed you, when you put them together, and that they may respond. And so I think it's the definition of antigenicity really needs to be reevaluated.